Okay, now we're recording. So this, I'll just repeat a little bit of what I said so for the benefits of the record. So this column is carcinogens, and those are factors that can either initiate or promote cancer. This is the process. So what we're looking at is factors that may cause a genetic change that are called oncogenetic factors. We're going to talk more about those in a few minutes. Radiation can cause irreversible DNA damage. So can some viruses and some of the chemicals we find in a cigarette. As a result of repeated exposures to these carcinogens, we increase the possibility of irreversible mutation of DNA. And that brings us to step number 2, promotion. So additional changes to DNA and cell structure, plus replication, increased mitotic divisions, and so on, is what we're talking about when we talk about promotion. Factors that are involved with that are some of our food additives, some of our industrial chemicals, and so on, that can be described as promoters that act on the DNA change cells, would not act as promoters on a normal cell. The change in the cell down this column looks like this. We're going to see a normal cell that's exposed to radiation or exposed to an oncogene that becomes a mutant cell. The DNA has changed. We get some cell division of the changed cells. Possibly this process will be interrupted by some genetic factors that are immune oriented, designed to destroy the cell before it gets a chance to proliferate. We're going to talk a little bit later about some of those things. And if that's missed, then we're going to get proliferation, and we're going to start to see a cell that takes on characteristics of an injured or damaged cell, an abnormal cell in presentation. Ultimately, that abnormal cell will lose differentiation and proliferate, increase in numbers. Two factors that represent part of the genesis of a cancer: loss of differentiation and promotion of numbers of cells. Again, there's another step here that may be recognized by the immune system. We hope so. And like I said before we started recording, it happens to the tune of billions of cells per day that undergo mutations somewhat as an expected event or occurrence, and are dealt with by our immune systems in multiple ways, either by signaling that goes on within the changed cell or from other cells in our immune system that recognize the changed cell as a non-self cell. So DNA damage represents initiation, proliferation under the influence of growth promoters. We're going to talk about some of those in a minute. Represent the next step, promotion, and then the development of a cancerous phenotype. What that refers to is that when we get to the point where the increased numbers of cells are starting to create symptoms and present as a pathology, we're going to be able to see features of that pathology that give us this phenotype. It progresses to the point where we'll be able to recognize that it was originating in a breast or in a lung. Perhaps it metastasized and presented in other locations, but the phenotype is going to give us the original tissue or cell type that the mutations developed from. So that's what we're talking about when we say we get a phenotype, a cancerous phenotype in that respect there. Okay. Oh, this is just an illustration. We might as well stay with our illustrations here that describes two different visuals that give us a visual of metastasis. So we can see that we're going to have a primary tumor, and the primary tumor is identified here and here on the two diagrams. We're seeing mutation that involves some angiogenesis, in other words, blood vessels that are close to these mutated cells. Proliferation, the cell numbers increase. Invasiveness, that means that we're breaking through the basement membrane of the tissue, whatever it might have been. These little blood vessels are describing our angiogenesis here. Now these cells are able to infiltrate the blood vessel and spread to distant locations and become a metastatic tumor, a growth, a cluster, a colonization of cells at a distant site. Same picture really here. This is a description of the cells that enter either the lymphatic vessels or the blood vessels and are able to travel, in the case of the lymphatics, first through a lymph node where they may be destroyed by cells located in a lymph node or immune system. If they're missed, they'll carry on into the circulation. Remember, lymphatic vessels empty the lymph back into the cardiovascular system in the interest of blood volume homeostasis. So ultimately, the lymphatic vessels will lead back to the cardiovascular system and able to spread through the cardiovascular system and nest or colonize in sites distant from the original tumor, like multiple tumors in lung tissue, very vascular tissue, so good chance for colonization to occur. In this diagram here, what we're showing is that the primary tumor was from breast cancer, and that we've got secondary locations for tumor formation, in this example, in the brain or in the lungs, in the liver, and in the ovary. So these are examples of metastatic locations distant from the site of origin. So they followed through lymphatic vessels associated with the arm or the axillary nodes, big cluster of those, is so very common mode of metastasis for breast cancer because of the proximity to those lymph nodes. And then circulation through the blood to get to some of these other distant sites here. Okay, so those are some good diagrams to refer to just to illustrate some of those features. We're going to get to that one a little bit later. Okay, now what we want to get onto is understanding a little bit more about how cancer forms. So when we talk specifically about carcinogenesis and the formation, we've already established in multiple ways that it's a multi-step process. The multi-step process makes it complex, makes it difficult to present, and also difficult to treat. So that's the real challenge behind cancer generally. So cancer, we know, is primarily an abnormal gene expression. At diagnosis, there's already billions of abnormal DNA cells, and some of these are known as driver mutations that will promote proliferation and promote malignancy. So let's see here. How do we want to summarize that? So cancer is initiated by abnormal gene expression that also involves proliferation so that at diagnosis, billions of abnormal cells are in existence. That's a pretty good way to summarize what we were saying. So some of these abnormal DNA mutations are described as being driver mutations. Driver mutations promote malignancy and promote proliferation. So not just spread, but numbers. And some other types of mutations are described as passenger mutations. So you can see by these two terms that this one is going to be less active towards the formation of cancer. So a passenger mutation has no real role in tumorigenesis. It's a mutation, so it's a change. It's probably going to represent loss of function, but it may also be more likely to be a type of benign tumor or a tumor that doesn't have a direct effect on the overall health of the individual that's been exposed to this type of passenger mutation. So keep in mind that because we're talking about abnormal gene expression, there's a number of ways that this gene expression can become abnormal. Remember, I said earlier today, we have 23 pairs of chromosomes. There's 20 to about 25,000 genes made up of DNA pairings. Many of these pairs are in addition to the genes, the 20,000 to 25,000 pairings that represent genes, there's billions of base pairs that represent total DNA. Probably somewhere in the vicinity of about 3 billion DNA base pairs exist on the 23 sets of chromosomes that we have in each human cell that has those 23 pairs of chromosomes. So lots of ability for mutation, lots of ability for signaling. The billions of base pairs that are not genes are part of what we call the epigenes. They're outside of the genes, they're around the genes. Sometimes they're called introns, but we know what they do is they have a direct influence in many cases on gene expression. They don't become genes, but they can influence gene expression. We used to think it was all junk DNA, meaning that we didn't really see that there was a function or anything. But the more and more we've learned about the human genome, the more we recognize that those genes have a purpose, but they influence gene expression. So there's a category of genes involved with cancer. And when we talk about these genes, we're really talking about oncogenes or oncogenesis. So these are coming from proto-oncogenes. Now, proto-oncogenes are normal genes. If we lose the proto part through mutation, they become oncogenes. Oncogenes are cancer-causing or cancer-promoting genes. Okay, so oncogenesis is going to be the result of this specific pathway, the specific pathway of creating cancer from a genetic origin. So these are genes that contribute to cancer if mutated. And when that happens, they're described as gain of function mutated genes. In other words, they're going to promote growth. So these are growth activating genes. Growth activating is another way to describe the same thing. Gain of function, growth, promoting proliferation. So if the proto-oncogenes become overexpressed or they mutate, they're described as oncogenes. And when they are described as oncogenes or become oncogenes, then they're going to promote growth in a cancer. They're going to promote proliferation. They're going to promote mitosis, and the cell numbers are going to start to be increased. So there's another group of, so there's these proto-oncogenes, is one group of genes. So basically they represent a genetic risk factor. As long as they remain proto-oncogenes, they don't have an effect on cancer. But if they are mutated and they lose the proto descriptor, they become promoters or gain function for cancer. Okay, now to counter those, we have a category of genes that are called tumor suppressor genes. Tumor suppressor genes do just what it sounds like. Normally, they inhibit cancer formation. They inhibit cell proliferation, cancer cell or any cell proliferation. So these would be also known as loss of function We had gain of function. Now we have loss of function mutations. So their role, their function that we're referring to in this case is that they inhibit cell proliferation. If they are mutated so that they no longer perform the job they're supposed to do, where they've lost their function, and as a result, proliferation is going to be allowed. So what we're looking at is cancer is more often than not So cancer is often a combination of mutations to both 1 and 2. If 1 doesn't mutate, we don't get proliferation. If 2 doesn't mutate, we get suppression. You follow me okay with that?
Yeah.
Okay. So basically what we're saying is if one of those things happen, we're probably not going to get cancer because the other one counters it. If both of them happen, we mutate both so that we get gain of function with respect to proliferation and loss of function with respect to suppression. Now we got cancer. And we're going to find out a little bit later that even these two things don't guarantee a cancer. But that's why we say cancer is often a combination of both mutations to both the tumor suppressor genes and a group of proto-oncogenes. So let's look at a couple of examples. Some of them I think you might be familiar with. Some of them maybe not. Some of them you've maybe heard of before, but not realized the role that they might play. So when we look at some examples of proto-oncogenes, so some of our normal growth factors that occur in normal metabolism, normal metabolic activity throughout the body are examples of proto-oncogenes. These would be categorized generally as growth factors. So you've probably heard of some growth factors. There's something called platelet-derived growth factor. It's PDGF, platelet-derived growth factor. There's something called EGF, epidermal growth factor is EGF. There's something called transforming GF. So that's just that would be a TGF. And sorry, let me just make some space there. There's subtypes of this type, so this would be an alpha. So it would look like TGF alpha. So these are just some examples of some different types of growth factors that fall into this category of being proto-oncogenes. So under normal conditions, there are cell-to-cell signaling that promote cell size and numbers, will promote regular mitotic divisions, and these local cells will release growth factors that bind to receptors on nearby cells that activate cell signaling inside the cell, that second messenger mechanism that we talked about in the first lecture. So in this case, what they would do is promote proliferation, or we may have some inhibitory factors that would inhibit proliferation, but this would be normal cell activity under normal conditions. Now, if those growth factors are overproduced because of mutation, so we're just going to say involved in normal cell signaling for growth and mitosis, proliferation, okay? If they are overproduced because of mutation of the proto-oncogene, they can produce excessive growth. Okay, so these are small peptides that can't cross the cell membrane, but they can cause intracellular activation of growth factors. So they encourage the nucleus to undergo mitosis. So when they're overproduced, the result is going to be an excessive, often self-stimulated growth. So self-stimulate means we've lost some negative feedback. So it's going to continue to ignore normal checks and balances as far as that goes. So the cell signaling becomes aggressive. So that's a scenario where we produce excessive growth factors because of the mutation. Or we can have mutations that affect growth factor receptors. So oncogenes mutate, and the result is we produce new receptors. Now, remember, one of the definitions of a receptor is that they're specific. So sometimes in cancers, the receptors become less specific. So they may be more receptive to other factors that will cause different types of growth and abnormal responses in the cell. So that's what the significance of a new receptor might result in. The other possibility is that we could get upregulation of those receptors. So that happens on a normal basis throughout the body at any time. So basically receptors that are specific, in this case to a growth factor, if the cell is required to grow because of regulating messages that are suggesting that that should happen, one way we can ensure that the cell grows is to make more receptors available to that growth factor. That's called upregulation. So the cell can insert more receptors. If we want to slow down growth, it can remove those. That would be called down regulation. So with respect to receptors, upregulation and downregulation is a mechanism that's utilized by many cells to help regulate normal growth and development. So in this case, we've got oncogenes that are signaling the receptors to upregulate even when we don't want it to. So, and the result of that is that there's more receptors available even if the growth factor doesn't change in terms of amount. So even if the growth factor is bound or not being produced, the small amounts that are out there are going to be more likely to bind if there's more receptors for binding. So that's what I'm talking about when I talk about upregulation. And the third mechanism that can happen with respect to changes to these receptors is that we can create receptors that have an increased affinity. To the growth factor. In other words, they increase their sensitivity to the growth factor. They may have a chemical attractant that is released to specifically take care or to particularly draw that growth hormone towards that receptor. So any of these changes increase the responsiveness to these growth factors. And as a result, we're going to get an increase in mitosis, an increase in size, and so on. You following me okay with that?
Yes.
Okay. Now, the other thing I'll remind you of, I don't think I've reminded you guys yet. I talked to the other group about this. You notice that I put 3 options down here. So when there's 3 options like this, it's a good idea when you're reviewing your notes and so on to think about what that might represent. And in a test, that looks a lot to me like a potential multiple choice question, where whatever number 4 is incorrect. So you might see a question that suggests that oncogenes can have effect on growth factor receptors in many ways. Which of the following would not be a mechanism that increases growth factor receptor activity through oncogenes, or something to that effect? These 3 that we've written in, those would be correct answers. You're looking for the 4th one, right? So anytime you see little groups of 3 or possibly 4, you should be thinking when you're studying and summarizing your notes and so on, that where can I group some stuff that might represent a question, a potential question? And now I've got an answer. I've got a good way of thinking about that particular group of answers or group of features that we've been talking about. Does that make sense?
Yes, thank you.
Yep.
So we've got an example here that's a fairly common one. It's called the human epidermal growth factor receptor type 2 gene. Okay, the abbreviation for all of this is HER2. You don't even see the dash. A lot of times I'll take it out because most of the time you're not going to see that. It's just HER with the number 2 at the end of it. HER2. When this receptor type is overexpressed, it's going to be seen in a number of cancers. In other words, what I'm trying to say there is that in certain types of cancers, this receptor type is overexpressed. It's upregulated, in other words. So in a particular type of breast cancers, they are described as being HER2 positive breast cancers, and that occurs in about 10 to 15% of all breast cancers. And so part of the carcinogenesis of this type of breast cancer is overexpression of HER2. Okay, and we also see that in some types of stomach cancer and esophageal cancer, about 30% of them are overexpressed with HER2. So when you see a cancer described as being HER2 positive, what we're talking about is a receptor, a growth factor receptor that is being overexpressed. So that means it's going to present as one of the 3 mechanisms that we listed just above. So what we're saying is 10 to 15% of breast cancers and about 30%, so about 30% of gastroesophageal cancers are HER2 positive. So the mechanism that we recognize that is part of the carcinogenesis, part of the stepping that's going on, is genetic alterations that result in an upregulation of these particular receptors that are causing proliferation, causing growth of these abnormal cells. Okay, so we're talking about mechanisms that lead to genetic mechanisms that lead to cancer. Okay, so now we've talked about some growth factors, we've talked about some receptors. We're going to look at a couple of other mechanisms here. I got to add some layers. Okay, so some other mechanisms that can lead to cancer formation are factors that affect the signaling pathways inside the cell. Remember when I talked about the second messenger system? So here's a receptor on the outside that would be specific. Okay, so this represents our first messenger. It activates a mechanism inside the cell that ultimately is going to signal the nucleus of the cell to cause the nucleus to release messenger RNA that binds with ribosomes to create the function in that cell. If the first messenger binds with the receptor and causes intracellular or cytoplasmic signaling to occur, we change the behavior of that cell. Right? That's the way some of these hormones work. That's the way other messengers that are too big to cross the cell membrane are going to work. So the intracellular or cytoplasmic signaling that we're talking about are the blue lines there, what I'm talking about inside the cell. Cytoplasmic or inside. So if they are altered genetically, the signaling for those pathways are altered, then they can cause the cell's behavior to change, to cause that cell to begin to proliferate as well. So an oncogene, in this example, an oncogene can cause proliferation by increasing or changing, let's the intracellular pathway. So we have enzymes and chemicals that are part of this pathway. They're going to change in the way that they signal a cell. Normally, they would transmit signals from the activated cell surface. That's what we showed here. So normally, they activate in response to first messenger receptor complex, right? That's what initiates the intracellular pathway. When they're mutated, but in abnormal scenarios, they code for an increase or an abnormal activation or proliferation. So we're going to activate an abnormal response. And that typically might respond by having a proliferation of abnormal cells, increased number of these abnormal cells. So there's a category of genes, they're common. They are ARAS genes. That's a family of genes that are found pretty much in all cells that synthesize proteins for normal cell signaling. So they would be responsible for regulating mitosis, maybe regulating the formation of hormones like insulin, if it's a beta cell in the pancreas, and so on. And they promote normal growth and function of a cell. If that gene is mutated, then it's going to promote cancer by promoting growth and spread. So a mutated gene for RAS is going to promote, so that's an example, normal growth, and let's just say cell signaling. But if it's abnormal, we're going to end up with cancer via proliferation and spread, malignancy. In other words, when we say that word spread. So, for example, somewhere in the vicinity of 15 to 20% of all cancers have the abnormal RAS gene. So 15 to 20% of all cancers in humans have abnormal RAS. I shouldn't use a capital. It's traditionally all lowercase, so we better stay accurate there. RAS. So again, I'm giving you some percentages because they're, just from a reference point of view. I will not ask you questions about these kinds of percentages, except in very specific scenarios. If it's a very, very important thing for you to remember. I don't want you to waste your time on trying to remember numbers. I want you to focus on the concepts. So we're talking about genetic mutations, and we're talking about the location where those genetic mutations have an effect. So we started with growth factors, then we started with some receptors, and now we're looking at some that are inside the cell. Okay, so we've kind of got some examples along the way. So I said that 15 to 20% of all cancers in humans have an abnormal RAS gene. So that's going to be a factor associated with proliferation and spread. And that's what I want you to get from this example. I'm not going to ask you a question about the percentage of all cancers that fall into that category. Like earlier when we had 10 to 15% of all breast cancers are HER2 positive. I'm not going to ask you about that percentage, but I might ask you what HER2 positive means in a cancer. So you need to be able to tell me that that's a receptor gene that is overexpressed, making growth factors bind to it more readily and therefore promoting growth, proliferation. Does that make sense? You okay with that?
Yes.
Yes, thank you, Professor.
Okay, so there's another category here. I just want to mention it briefly. And these are called transcription factors. So we went from outside the cell to the cell membrane to inside the cell. When we talk about transcription factors, we're now inside the nucleus. So transcription occurs in the nucleus of a cell when the DNA transcribes to a messenger RNA. The messenger RNA is released and binds to ribosomes and synthesizes proteins in a functioning cell. So this transcription is going to result in normal cell division and growth and death of a cell, as well as cell functions. So the messenger RNA is read by the ribosomes to synthesize proteins specific to that particular cell. If we have a scenario where in the proliferation pathway, we could have growth factors, we could have receptors or intercellular mechanisms that promote proliferation. All of these would contribute to transcription in the nucleus. So let's summarize this. If proliferation factors include one, growth factors, two, receptors, and three, intracellular pathways. Okay, if proliferation involves those things, all of these all contribute to transcription in the nucleus of our cell. So normally these transcriptions factors are fairly tightly regulated to create a tight sequence of events that are going to ensure proper messaging between the DNA and the RNA and ultimately out to the ribosome. So if we have oncogenes that alter the transcription factors, we're going to change the way that the nucleus is going to be able to transcript. So we may have overexpression or inhibition of normal nuclear mechanisms. So that's what's happening when we talk about changes to the transcription. So there's other factors that will be described as transcription factors that keep normal transcription pathways in check. And they have some of their own sort of abbreviations and so on. We're not going to name them because it gets even more complicated as far as that goes. But keep in mind that when we talk about transcription factors, when they work normally, they ensure that transcription occurs in a regulated fashion so that nuclear activity between DNA, messenger RNA, and ultimately ribosomes results in the protein sequencing that we want in that cell, results in normal mitotic activity, normal growth and development activity. If we have a gene mutation, a proto-oncogene that becomes an oncogene affecting transcription, that's all out the window. And as a result, we're going to have, by definition, a mutation that results in proliferation, typically, and then ultimately is going to be described as a cancer. So really, at the end of the day, we've got 4 locations to consider where these genetic mutations can occur. On the growth factors themselves that are outside the cell, on the cell membranes that are related to those growth factors, intracellular pathways that the growth factors activated, and then transcription factors that the intracellular pathways activated. That's why we say the factors that affect the nucleus come from 1, 2, and 3 there: growth factor, receptors, and intracellular pathways. So keep in mind that at the end of the day, these proto-oncogenes are normal. They become oncogenes when they mutate. And when they mutate, they cause abnormal proliferation. So these oncogenes could be introduced to the body by a virus. They could mutate during mitosis within a cell. And there could be chromosome error or extra copies of proto-oncogenes known as amplification that can result in increased numbers of any one of these steps, depending on which proto-oncogene is actually affected as far as that goes. Okay, remember with viruses are not complete cells, so they require invading or hijacking a host's DNA mechanism in order to survive. So typically what they do is they inject their genetic material into the cell, if not outright become endocytosed inside the cell. But either way, the genetic material is introduced to that cell. It's going to interrupt transcription within that cell with the goal of replacing transcription so that the virus now will be replicated inside the host cell. Some viruses carry oncogenes. So that's the way the oncogene could be introduced to that cell. So you've heard of Epstein-Barr virus. That's a virus that's known to cause specific types of cancer. The papilloma virus is known to cause cervical cancer. And this is the mechanism that it's going to utilize in order to cause that specific type of cancer. Now, it's not all bad news. Not all of those genes were proto-onco, right? We talked about the fact that we have a tumor suppressor gene. So we want to talk about one in particular, the tumor remember, we said the tumor suppressor gene is one that if we lose its function, we lose its ability to suppress. So it does just what it sounds like it's going to do. It's going to inhibit growth, inhibit proliferation of a tumor when it's healthy. So to become cancer, an important part of proliferation is inhibiting pathways that become abnormal. So these pathways are signals that would cause suppressor genes to become active. So if there was an oncogene that caused growth proliferation, these tumor suppressor genes would be activated to counter that, to make sure that the cells will not proliferate to the same degree. Now, they're tricky cells to study. There's tricky genes to study because they're only going to be expressed in the presence of cancer. So they're they're not going to be expressed if there's no cancer present. So if their absence is what causes some cancers, and so if you're studying a cancer that might be partly caused by abnormal suppressor genes, they're not there by definition to do their job. So it's really hard to study them and understand exactly what they do. But basically, tumor suppressor genes, if they both have to mutate. Remember, for a gene, they have a contribution from a mom and a dad to become an expressible gene in the progeny, in the child. So there's two parts to the gene. So typically both have to mutate for a tumor suppressor gene to stop functioning properly. So when that happens, the role of that tumor suppressor gene is going to be lost. The best example of a tumor suppressor gene, and I found out from the other class that you did not talk about this in your 400, according to them. Maybe you would say differently, but there is a protein out there called P53. This is an example of a protein that is directed by a tumor suppressor gene. So it's normal in low amounts of a healthy cell. And it's in large amounts in a mutated cell. So normally we would expect to find low amounts of P53 in a healthy cell. Now, a lot of it is stored right in the nucleus, and a little bit might be circulated in the cytoplasm of the cell. So this is right in the location where the mutation is going to be occurring, right in the chromosome material that's located in the nucleus. So P53 is expected to be found in low amounts if the cell is healthy. It's expected to be found in large amounts in a mutated cell. So this is the most common defect of tumor suppressor genes in cancer. So it's the most common defect in tumor suppressor genes that lead to cancer. So somewhere in the vicinity, plus or minus 50% of all cancers has abnormal p53. So this p53, when it's working, has been nicknamed the guardian of the genome. And this is why. When p53 is activated in an abnormal cell, the results are going to fall into three categories. What does that sound like? What's that starting to look like?
A multiple choice question.
There we go. Yeah, that's what I'm talking about. So we expect it in low numbers, but in a cell that has DNA damage, it's going to be in large numbers. And there's three main functions that it has responsibility for. So p53 will bind itself to the damaged DNA, binds to the abnormal DNA, and tries to repair. So it's trying to repair the damaged DNA. Another factor is that it will slow down or interrupt mitosis. If we interrupt mitosis, we buy some time for repair. We don't allow mitosis to continue on and create two daughter cells with the abnormal DNA in it. Okay. Now, if those first two things can't work, if the damage is too significant or there isn't enough time, then the third responsibility of p53 is to initiate apoptosis. Now, I mentioned this before. Hopefully, you talked about it in 400. Apoptosis, a nice, simple, clean definition for apoptosis is that it is pre-programmed or programmed regulated cell death. It's neat and tidy. It happens all the time. Earlier today, I said to the tune of billions of cells become abnormal in a given day during mitotic divisions and all that sort of thing. So this p53 is active all the time. That's why it's nicknamed guardian of the genome. So what's going to happen is a chromosome aberration or mutation occurs, and one of these three things is going to happen. It can't repair, and the time gained by the interrupted mitosis is not effective, then we're going to get apoptosis. So this works most of the time in an otherwise healthy individual and really represents a type of an immune response by a chemical, in this case, p53. So I've got a diagram I can show you that will summarize that for us. Let me get there. Okay, in this diagram, what we're looking at is three scenarios. So we've got DNA damage in an otherwise normal cell with low amounts of p53. So p53 is described here as being normal. There's DNA damage, p53 is activated and binds to DNA. Okay, so the goal here is to try to repair. If it's successful, we've got normal cells. If the repair fails, we've got apoptosis. Okay, so we got, in this case here, the two things that are happening before we get successful repair is inhibition of mitosis and DNA repair. And if those things aren't working, apoptosis. That's what we expect with normal activation of p53. If we have a cell with mutated p53 and we get damage, we don't get activation of the p53. We don't see an increase in the amounts of p53. As a result, no repair, no senescence, which is the fancy word for interrupting the cell cycle, no cell cycle arrest. And as a result, the mutant cells are allowed to continue to reproduce, and we get malignancy. This is just another pathway that's involved. Now, before we leave this particular topic, how much time are we dealing with here? We're winding down, and I'm not as far along as I want it to be. Okay, so before we finish this particular topic of tumor cell suppression, I'm going to mention one other one. Okay, let's go. So here we're in our tumor suppressor gene. We're talking about p53 and some of the pathways that it's involved with. Some other examples there, and you may have heard of these ones because they're in the news quite often. There are two other genes that are expressed in humans. They're abbreviated as BRCA1 and BRCA2. They're called BRCA because they're tumor suppressors that normally are important with respect to breast cancer. So normal functions of BRCA1 and 2 are important for DNA repair, particularly in breast cancer. So they normally promote DNA repair. Okay, and specifically BRCA1 also promotes p53. Okay, so not only do both BRCA1 and BRCA2 promote DNA repair, BRCA1 also promotes p53, so it's kind of like a positive reinforcement. We're going to bring in some p53 as well as do our own business as far as that goes. So in a family, a female with a family history of breast cancer and a defective BRCA1 gene, that person hasn't, so if our female has a family history of breast cancer and abnormal BRCA1, 50% increase risk of breast cancer. Not only that, it will, has an earlier age of onset and a higher risk to be bilateral. So much more aggressive as far as that goes. So what we're saying is if you have a BRCA1 gene that's mutated, your risk for cancer is 50% higher than someone who does not. And the age of onset is earlier, and breast cancer is more likely to be bilateral. So this inherited breast cancer is not a common breast cancer overall, but when it is known to be inherited and the BRCA1 gene is mutated, that's the mechanism behind that earlier onset and the more likely to be a bilateral presentation. Abnormal BRCA1 and 2 also increases the risk for other cancers, like ovarian or prostate and pancreatic. So it's not just about breast, but it is definitely an implication in breast cancer. When we talk about breast cancer, we'll talk about breast cancer that can be described as being HER2 positive, as well as BRCA1 and 2 positive, a triple threat as far as risk factors go in that respect. So the HER2, remember, that was a receptor gene that became defective and attracted more growth factor. and the BRCA1 and 2 defects interfered with normal DNA repair as far as that goes. So when we talk about the formation of cancer, typically activating an oncogene or inhibiting a suppressor gene is not enough to cause cancer. Not enough to lead to cancer. There needs to be more steps. So several steps are required. The steps can be summarized as initiation. We showed you these, I think, earlier. Promotion. And proliferation. This is another 3-step. Now, there's many steps sometimes within these steps, but these 3 things also look like a pretty good multiple choice question. So when we talk about initiation, we're talking about factors that cause a genetic mutation. So we've activated the oncogenes or we've inactivated a suppressor gene, but when these changes are not detectable. So this part of cancer development, carcinogenesis, this part is often undetectable. So this is any mechanism that results in DNA mutation. So we said we could activate oncogenes or inhibit suppressor genes. That's what's happening during initiation. And that's typically difficult to prevent, undetectable. We don't know what's happening out in general society with respect to exposures to carcinogens that are described as initiators. We don't know how much sun exposure you've had or what that effect might have on those oncogenes. Some people can stay out in their sun in their entire lives and never activate an oncogene that causes the DNA mutation. And other people are more likely to see that develop into a genetic change. So when we talk about promotion, what's happening during promotion is, oh, I don't want proliferation in here. I want progression because we're going to use that. So we want proliferation right here. If we're going to be able to promote the mutated cells, that's represented by the word proliferation. That's mitosis. And that means we're going to increase mitosis outside the normal checks and balances. We probably need several mutations and in order for this to develop, right, based on multiple exposures and so on. And the consideration here is that if the cell is a non-mitotic cell, they're not as likely to cause cancer. So, for example, up in the brain, in the central nervous system, the neurons themselves are less likely to ever develop into a cancer than the supporting cells, the neuroglial cells. When you hear about a cancer in the brain, one of the more common ones you'll hear about, for example, the lead singer for Tragically Hip, he had a neuroglial blastoma. glial cell tells you it's one of the support cells. It's not a neuron. It's the, you know, the general public would call it a brain tumor, but specifically it's one of the support cells within the nervous tissue that's undergoing rapid mitosis and creating a mass that puts pressure on a part of the brain. So the symptoms in that case are often as soon as they create enough cells to cause pressure on a zone in the brain that has a particular responsibility. So we'll find out a little bit later when we start talking about the central nervous system, you can put a pressure on a certain part of the cortex that affects the field of vision. So the first thing the person might notice is they've lost visual recognition in the left side of their field of vision, for example. Or when they look up, they see nothing. Or when they look over to the left, they can't see anything. Now, if they turn to the left, because the other parts of the eye can then see, that's when they're looking straight ahead and asked to rotate their eyes to the left for field of vision that they can't see in that left side. It's an indication of where the pressure is being applied in the brain. So for proliferation, we got to have several mutations that result in proliferation, in mutations that give us an increase in mitosis. Remember, carcinogens are anything capable of causing cancer. Some are described as initiators. They're the ones that cause DNA damage, initiation, and some are proliferators. They stimulate growth. A complete carcinogen can do both. That's what a cigarette is. When we talk about other parts of promotion now, proliferates. Let me just make sure we've talked about that. We know other factors that might promote proliferation. We know that when you hear about it this time of year, you hear about people saying, well, you know, barbecue, if you char your meat on a barbecue, that presents carcinogens. When you eat it, you increase your risk for carcinogenesis. Some certain food additives, food colorings, and different things increase carcinogenesis. This is the stage where they have their most likely effect, not on the actual DNA mutation, but more on the proliferation step as far as that goes. We know that some hormones are promoters. I mentioned estrogen earlier. It's a promoter for breast and ovarian and uterine cancer. If a woman has had more menstrual cycles in her lifetime, that means she's had more exposure to estrogen. She's at an increased risk for breast and ovarian and uterine cancer. Okay, when we talk about factors affecting progression, that's when we get to progression stage, we're really talking about malignancy. So in this step, what we're seeing is that initiation is not enough to cause malignancy, but proliferation of unstable cells can cause more mutations and growth advantage over normal cells. Remember, we said that some of the ATP is redirected towards the abnormal cells over the normal cells. So in progression, we're talking about malignancy. So proliferation of unstable cells can cause more mutations. So you think about it, you've got an unstable cell, it's an abnormal cell because it was mutated. Now it's being proliferated. Mitotic rates are higher. There's a much more likely occurrence for further mutations to occur because you've got more rapid mitotic division going on. You've got more rapid changes in chromosome arrangements that are likely to occur with the speed of mitotic division going up. More proliferation means more mutation. We create new cells that are very different from the original ones, the cells they developed from in the first place. So that chromosome presentation can be very strange as far as that goes. So there may be extra chromosomes, there's missing, they're in the wrong place. But the point is that through these steps, initiation, promotion, progression, there are many steps that are required in order to get to malignancy. Many possible interventions can occur. We can intervene with our P53 at the level of DNA mutation, along with supporters like BRCA1 and 2. We can intervene at promotion with recognition from immune cells that might find these cells clustering or accumulating in a particular location, and recognition will lead to activation in the immune system. Preventing initiation is difficult, but repairing is possible, and interrupting proliferation is more possible. And those things, preventing promotion and progression, is going to lead us to preventing malignancy, and that means treatment is going to be easier. If we can get to treatment before malignancy has occurred, treatment and outcome prognosis is much, much better as far as all of that goes. Okay, I think we need to stop there for your sake more than anything else.